All right, and thank you very much, Jamie. As you heard, my name is Laura Tibbs Cortes, and I'll be presenting on the status and prospects of GWAS in plants based on a review I recently published. Now, I know a lot of you are already very familiar with GWAS, maybe used in your own research, but just to bring everyone up to the same page, I wanted to talk just a little bit about what GWAS is. Well, it stands for Genome-Wide Association Study. And the goal of GWAS is just to detect loci where variations in genotype are associated with variations in phenotype. So just for a little toy example of how GWAS works to show you how that does work, first you start with a group of diverse lines. So let's say that in this case, we have inbreds A, B, C, D, and E. And we also phenotype all of them. Again, simple example, let's say that we're just looking at height and we're calling them either tall or short. Next, for this genome-wide association study, we need genotype information. So let's say we got the genotypes of these inbreds at 3 SNPs. Now, as I said, the goal is to look for loci where genotype variations are associated with phenotype variations, and we see that happens at SNP 2, where if it's a T, the phenotype is tall, and if it's a G, the phenotype is short. Again, simple example, but in general, even the most complex models are doing something like this. Then GWAS results are shown on something called a Manhattan plot, which apparently someone thought looked like the skyline of Manhattan. Along the x-axis, you have the chromosome numbers, and the y-axis, the negative log of the p-values. Each individual dot represents a single SNP, and its height shows how significantly associated that SNP is with your phenotype of interest. So here we can see there's one nice big peak on chromosome 4. So whatever this trait happened to be, there's something in that area of the genome that's affecting this trait. Now, so we talked about what GWAS is, but really, where did it come from? How did it develop? Well, around 1994 to 1996, there were several early papers that proposed association studies. These couldn't quite be genome-wide association studies yet, though, because they simply didn't have genome-wide data. In 2001, though, we had the publication of the draft human genome and some early SNP data sets, so that we were able to have the first GWAS publication in 2002 based on that data and studying human heart attack risk. Finally, in 2005, about a decade after GWAS was first proposed, we had the first GWAS published in plants, in the model species Arabidopsis. And then, of course, from 2006 onwards to today, GWAS has rapidly expanded into a ubiquitous tool in crop species. There are so many applications of GWAS in crops today. For one thing, you can study a wide range of phenotypes. This includes things like your traditional agronomic or physiological traits, so your yield, your plant height, but also biochemical phenotypes. For example, Jamie mentioned that I work with vitamin B levels in maize grain, which I would call more of a biochemical phenotype that I study with GWAS. You can also look at endophenotypes, which are just any phenotype in between the genome and what we would normally call your final phenotype. So that's the things like the epigenome, the proteome, and metabolome. And of course, you can also use GWAS to study the output of high throughput phenotyping, which I know a lot of people in our department work very hard on. Once you have your trait of interest, there are many applications of GWAS itself. First, GWAS can just be a standalone analysis that can help you to understand the general genetic architecture of your trait in your given population, or GWAS can be a part of other analyses. For example, if you had a candidate gene that you were interested in, you could use GWAS to look at a particular population and see if that gene really is involved in the variation of that trait in that particular population. You can also use GWAS results in genomic prediction models or as targets for genetic engineering. So really, no matter what you're working with, whether it's a very molecular phenotype or high throughput phenotyping, whether it's a basic biology or really applied crop improvement, GWAS can probably be helpful for you. Over the years, a lot of work has been done to improve GWAS. Of course, there's a lot of detail here that we can't go into. So today, I'll just be focusing on 4 major improvements in GWAS, which are population structure and kinship control, multilocus models, setting significance thresholds, and efficient calculations. And over the years, in general, these improvements have worked to improve both the power of GWAS, statistical power, as well as computational speed, and both are important. First, why is population structure and kinship a problem? Why do we have to control for that? This is an example from another paper that I really liked and wanted to share with you all. For population structure, imagine that there are two islands. On the first, Abelia, the climate is very dry. At the A locus, we happen to know that the big A allele confers drought tolerance. And so big A has become fixed on Abelia. There's another island called Botania. This island is very wet and there's a lot of fungus. We happen to know that at the B locus, big B confers fungal resistance and big B has become fixed on Botania. Now, if we didn't know anything about this population structure, that there's actually two distinct subpopulations from two islands, and we just mixed these lines all together and did GWAS, we would find that both A and B loci are going to look like they're associated with drought, even though we know that in reality only A was biologically related, and that's due to uncorrected population structure. Kinship can cause similar problems. In the example of kinship, let's say that we have a diversity panel and a subset, this part in yellow, those lines are related. They're all descended from a cross between two of these Botanian lines. Now, remember, Botanian lines have low drought tolerance and are fixed for that little a, big B genotype. So we have this family within our larger panel. If we don't correct for that family relationship, that kinship, we're going to find that both A and B loci again look like they're associated with drought, even though, as we said, only A is biologically related. So both kinship and population structure can lead to these false positives. The mixed linear model is a solution to this problem by accounting for relatedness at both levels. For population structure, it uses structure or PCA to quantify the population structure and includes covariates from those in the GWAS model. And then for kinship, calculates a kinship matrix from the marker data that is included in the GWAS model as well. Accounting for both of those levels of relatedness can reduce your false positives. And so since the development of this framework, many different GWAS methods have been developed, all based on the same general approach. Another improvement in the statistical power of GWAS came from multi-locus methods. Single locus methods, like what I talked about in that basic example at the beginning, just look at each marker individually and its association with the phenotype. Multi-locus methods, though, test multiple markers at once. The first example of this was the multi-locus mixed model, or MLMM. This is an iterative approach. First, you run MLM GWAS, then rerun GWAS, this time including the most significant marker from step 1 as a covariate. Then you run GWAS again, now including the most significant marker from step 1 and from step 2 as covariates. And this process just continues until the model detects that you've included an optimal number of covariates. Now, this is particularly helpful in increasing power in the presence of repulsion linkage. An example of that comes from sorghum height. DW3 and QHD 7.1 are genes that both control plant height in sorghum and are located very close together on chromosome 7. They are also in repulsion linkage. The plants, at least in the population we looked at, plants with height increasing alleles of DW3 tend to have the height decreasing allele of QHD 7.1 and vice versa. The result of this is that in MLM, single locus GWAS, we're able to detect some loci associated with height like DW1, 2, and 4, but DW3, as we can see when you zoom in on the area on the right here, is not significant. Nothing comes above that significance line. That's because any SNP that is linked to DW3 is also linked to QHD 7.1. If a particular allele of a SNP is linked to height increasing DW3, it's probably linked to height decreasing QHD 7.1, with the result being that they kind of cancel out. And so no SNP appears significantly associated with height. The multilocus methods can address this. We still detect DW1, 2, and 4, but now we can see DW3 and QHT 7.1. As this model iterated through, as soon as some of these other SNPs, particularly DW3, were included as covariates, we could see QHT 7.1 as well. So now, instead of detecting neither DW3 nor QHT 7.1, we can detect both. Setting an appropriate significance threshold is also important and a challenge in GWAS. This is because some common methods like Bonferroni and false discovery rate, or FDR, assume that tests are independent or that test statistics are independent, which is not actually the case in GWAS because of that LD between markers. Another option, permutation, can be very computationally intensive, especially with large data sets. One potential solution comes from simple M. This method estimates the effective number of independent markers in the data. It estimates that by the number of PCs, principal components, needed to explain at least 99.5% of the variation in the genotype data. Then the significance threshold is just set as alpha divided by that effective number of markers. Of course, there have been many potential solutions proposed, but simple M is a fairly computationally tractable one that also happens to line up quite well with the results of permutation. Here, using that same GWAS plot from our sorghum height example, you can see what a difference it makes to use different significance thresholds. The most stringent here is Bonferroni with a negative log p-value of 6.73 for its threshold. Simple M comes in next with 4.76 and FDR at 3.7. Though of course, as I said, simple M is probably of those three a better option due to not making those assumptions about the independence of tests. It's also really important to improve computational speed because GWAS datasets are constantly increasing in size. I know that personally with my own projects, I'm working with about 12 million markers and about 2,000 individuals. So that can take a very long time. Some solutions are just to have more efficient calculation. One method is through approximation methods, which give you almost the exact same answer as an exact GWAS approach, but because they use approximation, can reach that answer much more quickly, or more efficient implementations of previous GWAS methods. A good example of this was actually developed by Aaron in Dr. Schnabel's lab here at Iowa State, when he developed Farm CPU PP as just a more efficient way of running the Farm CPU GWAS method. Of course, there are also many Bayesian GWAS methods, though I've mostly been focusing on frequentist linear model methods today, but the Bayesian GWAS methods tend to be even more computationally intensive and therefore slower. So it's even more important for them to continue to increase computational speed to become more broadly useful. We can also apply what we've learned in GWAS in other areas, moving beyond GWAS. One of these ways is through OAAS, which stands for Omic-Wide Association Studies. Basically, in OAAS, you use GWAS methods to study associations between phenotypes and other types of omic data. An example I use in my own work is TiWAS, or transcriptome-wide association studies. In place of the genotype data of the GWAS, we instead use the transcript count from each gene. Here is on top a GWAS results and on the bottom TiWAS results from the same vitamin trait. You can see that there's a very strong signal on the TiWAS results that's directly under a strong peak in the GWAS. This can be really helpful. These two results inform one another so that since this is a strong TiWAS peak, it's telling us that, hey, what's going on under this GWAS peak has to do with the different transcription levels. It also can help us narrow down that peak because while GWAS tells us a genomic region, TiWAS actually picks out a single gene because it's looking at the transcript count for an individual gene. Genomic prediction and selection also have a close relationship with GWAS. Both use genotype and phenotype data, in the case of genomic prediction and selection from the training set, to estimate marker effects. Genomic prediction then uses these marker effects to predict phenotypes of the testing set, which are just unobserved individuals. And genomic selection applies genomic prediction results to make selection decisions in a breeding program. These similarities mean that GWAS methods can be adapted for genomic prediction and selection, and vice versa. And this has in fact been done. In addition, GWAS results can actually be directly incorporated into genomic selection or prediction. First, published GWAS results can be used and incorporated into a genomic prediction model, or if there are no published GWAS results on your trait of interest, you can use a technique called GS plus de novo GWAS. In this technique, you conduct GWAS on the training set. Then, significant markers from GWAS are used as fixed effects when predicting the testing set. There's also a synergistic relationship between genome editing and GWAS. On the one hand, genome editing enables validation of GWAS-detected loci in new genetic backgrounds. This can help us to validate GWAS results and methods, thereby informing method research. On the other hand, GWAS identifies loci that underlie traits, which of course provide targets for genome editing. Now, bringing this back to our general theme for this semester's seminar, we can think about GWAS as working through this big system where we have first the experiment, then the analysis, finally leading to understanding and application. In all of these steps, there are challenges to overcome, some of which we've been able to talk about today. But as we overcome these challenges throughout this system, this leads to more opportunities for that understanding and application, whether it's a more basic biological understanding or application for crop improvement. But really, after all, those are the goals. So overcoming challenges throughout GWAS really helps us to achieve those different goals. I'd like to acknowledge all my lab members and the funding and open up for any questions. Of course, if you think of more questions later, feel free to reach out or maybe even check out my review. Thank you.
Thanks, Laura. Does anybody have any questions for Laura? You can put them in the chat. You can unmute yourself, however you feel comfortable. Okay, well, I will get us started. I still have questions.
Okay.
Okay, first of all, a very simple question is GWAS was really conceived for human studies to look at human health. Is it still used for that? I mean, in your writing your review, is it still used for that?
Yeah, it's definitely still used for that. I mean, I focus more on agriculture related studies, but they're definitely still using it. Things like schizophrenia and things like that, there have been recent results in humans.
Interesting. I just know we often co-opt different techniques. And so I was just wondering if they were still using it or if they'd moved on to something else. Daniel has a question.
Sorry, I went from mute to, or I went from unmute to mute. Hey, Laura, thank you for that presentation. It was really good. And I did download your review. I've not read it yet. My question involves the OAAs, because I have heard of a TiWAS before. And so I'm wondering if you could expand in that a little more in terms of how often have you seen these different types of, different types of studies, I should say. And then for your TiWAS, if you could expand on, your data set and what type of sequencing did you use? Because my work uses RNA-seq, which is a really in-depth look into the transcriptome, but I was wondering if you used any other type of data set.
Yeah, so first, as for how many there are, things like that, I think from what I've seen that TiWAS would be the most common OWAS that I've seen. I still haven't seen terribly many of them. Like I said, I'm working on a project that has TiWAS. We're collaborating with some people that are working on another related TiWAS right now, and there's a handful of papers out using this, but not terribly many yet. As for the data set, In this case, for the TiWAS, we used a subset of about half of the lines that we use for GWAS, just because of the realistic, like, logistics of collecting that much RNA. And we specifically got RNA from 24-day-old kernels, and we used tag-seq rather than a full sequence RNA seq. Does that answer your question, Daniel?
Yes, yes, it does. Thank you.
Yeah.
Danny, do you want to ask your question? Then I'll read the one from the chat.
Sure. Thank you, Laura. It's so good to see that you have a paper published. Just wonderful. Congratulations on it. And it relates to this slide, actually. I was thinking, you mentioned you had 2,000 accessions and millions of data points or SNP points that you're using. And that's where things seem to be going in the GWAS world. People are continually expanding, and that's helping somewhat get a sense of overcome the issues of missing heritability and all that. But one question I have is there something called SWAS, which is combining information from multiple species, related species, and then running an association study? Because some of the underlying features would be common, but is it something are there softwares that are available to run an analysis like this, or it doesn't make sense to combine information across species to go into some of the species that are more closely related to expand the the scope of the study?
Yeah, I don't think I've ever seen GWAS across species. I think my biggest concern with that would be it would kind of be that population structure problem on steroids, kind of. You would get not just subpopulations, but actually different species. But presuming that you could deal with that in the way that we can deal with population structure, then I wonder if, so with combining GWAS and TiWAS, there's something called Fisher's combined tests that I've seen used, where you basically are using p-values from each method and combining them to get a combined p-value. So perhaps something like that could be applicable to combining methods across species, but that is not something I've seen.
Yeah, no, thank you. I was just thinking of some conserved regions if that would be beneficial. Thank you.
Yeah, thanks. No, that's a very interesting idea.
Okay, Clayton, I see you've got your hand up, but I think the chat question really goes nicely right after Danny's question. So hold on. The question in the chat, Laura, is, first of all, says, nice talk. Then asks, did you explore the use of sequential corrections, Hochberg and Hommel, or false sign rate in eBay's procedure for multiple testing correction?
Not, I'm trying to think about all the names you said. Not specifically. We covered, so in this talk, I only covered simple M. In the review, we mentioned, I'd say about 3 or 4 others. So there's definitely a lot of options out there. Those off the top of my head don't sound as familiar to me, but I guess the point is there's obviously a problem that different groups are working to fix, and that there's multiple potential solutions to that.
Excellent. Okay, Clayton, and then we've got another one in the chat.
Yeah, so great talk. Really appreciated the overview. It's really good to dig into a lot of those things. So really excited. In terms of actually executing the GWAS, like the logistics behind it, I'm fairly familiar with Tassel, like GEMA, Gapit, but I'm curious from your experience, are there other softwares or programs that you've got really good success with in terms of like 1, usability, or 2, like more in depth, but like, man, the results are a lot stronger, which I think would be more useful for us as we're digging into the genomes?
Honestly, I use Gapit myself. Okay. I think, yeah, so I mostly use Gapit. I think to me, the biggest improvements have just been doing things like using more efficient packages to read the data in, making like splitting the data up in efficiently to run on the server in parallel, things like that. So package wise, I think Gapit is great. It's more stuff I've done outside of that to make the running more efficient if you're working with really big data.
Okay, awesome.
Okay.
do you those are some skills that would be really useful. Have you categorized or compiled all of those outside things then as well?
Not super systematically, I guess.
Okay.
I actually had a kind of a guest lecture that I gave to one of Matt Hufford's classes about GWAS that I made kind of a mini GWAS tutorial that included some of those things.
Okay.
But that's not the review. That's just a little like coding tutorial, if that's.
Yeah, that's fair. I mean, if
If
there's
that would be-
anything, I love standing on the shoulders of other great people. So I would love to dig into some of that outside stuff that you've developed too.
Sure, if that's interesting, I can find that for you.
All right, thank you.
That sounds very useful. I think a lot of people would find that useful. Okay, Dr. Fay has asked, in open pollinated species for which inbred lines are difficult to obtain, what are the major challenges for using GWAS or OAAs?
Yeah, so like you just said, if it's hard to get an inbred, then it's harder to get, you know, genotype and phenotype on an individual, because with inbreds, we can cheat and use multiple plants that we know are going to have basically the same genotype and phenotype. But you can still do GWAS even if you don't have that. I mean, they're even able to do GWAS in animals where obviously you don't have inbred lines there either. Really, it's just if you have a lot of genetic variation between one individual and the next, in that case, you're going to be needing to have the phenotype and genotype both from that one individual rather than just from the same inbred line, but it's definitely still doable.
Muted myself. Okay, does anybody else have any questions?
Yeah, this is Asibi. I have a question. So I'm just looking at it's in regard to the TiWAS.
Mm-hmm.
You know, you said that is more specific to a gene, but I'm just wondering the situation where you might have, you know, two very closely related genes, or the situation where, you know, like one exon of a gene is, you know, attached to a related one and transcribed. How sure are we that, you know, it is the particular gene that you are looking at? You know, I don't know much about this area, but this is something I was just thinking of. So you might think it's gene A, but could it be B instead?
Yeah, okay. So something here is that this isn't always the case, right? That there'll be a single gene showing up in TiWAS under a particular GWAS peak, right? Mm-hmm. So over here, we have a GWAS, here's a significant SNP that has nothing under it in TiWAS. So that's telling me, hey, rather than being involved with transcription level, there might be something here with an actual different protein being produced, let's say. So this is a nice, simple example, as you pointed out, where it really does pick out a single gene under a peak, and that's not always going to be the case where it's going to be that simple. In the specific case of, basically, I think you're talking about kind of alternative transcripts where an exon of one gene is being, or two genes are so close together that you can get different transcripts. In that case, I think that's just kind of a larger problem with RNA type studies in general, that you need a good annotation of genes to be able to map your RNA to those genes. And if you don't have that, your results aren't going to be as useful.
Okay, thank you.
Thank you.
Yeah, I think my question was going in a similar direction. So with regard to TiWAS, I compared it to the genetical genomics approach where either cis or trans EQTL were detected. So in the TiWAS studies that have been conducted so far, which of the two types is predominantly found?
Well, as I said, there's not too much out yet, but it seems like there's a lot of that cis regulation that I happen to have an example of here, but that's definitely not 100% of the cases, right? It's just, I would say that's the majority.
I just have one question for Laura. Did you have time to check on mixed logistic regression models when you have non-normal data, for example?
Oh, okay. So for non-normal data, I did not specifically work with that in the review. Some stuff I know I've done in my own research has mostly just been transformation to get my data to be normal rather than using
Okay.
methods designed for non-normal data itself. So that's one approach.
Okay, thanks.
Okay, I'm going to cull the questions, but if you have more questions, hold on to them. And after our second speaker, and he does his questions, then I'm sure our speakers would be happy to stick around and answer more questions and we can have more of a discussion. So for now, thanks, Laura. Really appreciate it. I'm going to stop recording and I'll start again in a minute.